Hi, welcome to a special virtual presentation of the 2021 London Swine Conference. I'm Teresa Van Ray, your LSC chair and a very proud pork producer from Huron County. Last year, we marked our 20th conference. Since 2001, the London Swine Conference has helped connect producers, researchers, industry, and policymakers across our pork sector. It's an important opportunity to share science-based information that helps each of us do our part to make our businesses and industry stronger. So we all know that chatting with each other face to face and having that cup of Java, the odd glass of hops, or fermented grapes, my favorite, it's such a huge part of learning from each other and, well, a lot of fun. Remember, fun? Not sure about you, but I've not exactly felt in control of a lot in my life lately. However, knowledge is power. Knowledge can help us gain back some of the control we all need to have success. While we aren't able to gather this year in person, we are excited to bring you a series of live and on-demand presentations from people at the top of their fields. We are one of the many phenomenal industries that have continued to produce food for the world throughout this past year. For that, thanks to each and every one of you and your families. So, sit back. We invite you to enjoy this virtual presentation. And I, for one, look so forward to seeing each and every one of you face to face and celebrating with you in 2022.
Well, hello everyone, and welcome back to my virtual home office and the 2021 London Swine Conference video series. My name is Terry O'Sullivan, and I'll be your virtual moderator for this presentation. And before we begin, and on behalf of the London Swine Conference, I'd just like to acknowledge the support of our premier sponsors, Better Pork, Farm Credit Canada, and Total Swine Genetics. And with me today is Dr. Scott D. Hello, Dr. D, and welcome.
Terry.
Hello. Dr. D earned his DVM, MS, and PhD from the University of Minnesota, and he is also a board-certified microbiologist. And Dr. D spent many years in private practice, in academia, and now he is the director of Pipestone Applied Research with Pipestone Veterinary Services. And we are very fortunate to have him here with us to share his expertise on biosecurity and risks of pathogen spread and transfer. So welcome, Scott.
Thank you very much, Dr. O'Sullivan. I do appreciate the invitation to this conference, which I hold in high regard. I've been a participant as a speaker once many years ago, and I really enjoyed myself. And I wish I could be there today in person, like we all do. Wish we could, but the good news is that's coming. I just got my first dose of Moderna the other day, and I'm starting to feel more bulletproof as time goes on. So very excited about this presentation. You see the title. We're going to talk about hitchhikers, about pathogen movement. I'm going to focus on feed and what we do for that risk factor on the biosecurity side. But again, thanks for the organizing committee for the invitation. It's a real honor. I'd like to highlight my collaborators. This is a big team. This feed topic is interesting because it's almost 7 years old. Back in 2014, January, I discovered that PED virus was in feed bins in index cases consuming feed from those bins, and we recovered the virus from those bins. We fed it to pigs and showed that it was infectious. And since that time, there's been a lot of great work done by a lot of organizations here highlighted on this slide, but we couldn't do it without the funding so far. That's the Foundation for Food and Agricultural Research. Sam Nutrition is a company we work with on imports. We'll talk about them. The SHIC did a lot of this work on the funding side. There you see the universities that are involved. Eric Nelson from South Dakota State, Megan Niederwerder and Cassie Jones from Kansas State, Jeff Zimmerman, Iowa State, and then Declan Schroeder, Jerry Scherson, Pedro Aurelia, a lot of people at Minnesota right now working on this. Thanks to Beth Thompson, our director of the Board of Animal Health, and Gil Patterson from VetNow. You'll see some of Gil's work today, and then our group at Pipestone. So one thing I like about research is building teams, and I think we've really got a nice team here, and it shows, I think, in some of the work that's been done. So what I'm going to do today is kind of listed right here on our next slide is, first and foremost, just quickly review what's happening in China in regards to African swine fever virus, because everything really orbits around what happens in China. And China has many viruses that we don't want in North America, one of them being ASF. So we'll update that. We'll quickly review the science on feed, transport, and transmission. I don't want to spend a lot of time on that because I know you're familiar with it. And I certainly congratulate Canada for its efforts in developing a national program to manage the risk of foreign animal disease viruses through imports of feed ingredients from high-risk countries. So Canada led the way. It was March 29th in 2019, and I got the news from Doug McDougall that the national program was in place. I can't tell you how happy I was to see that based on the work we've done. So thanks for doing that. It's a great model for the rest of the world. I see Australia has followed suit. Now we need the US to do it. But what I'm really going to focus on today is 3 new experiments. I like to bring new stuff and show you some new things. And I'm going to show you some new experiments on viral survival in feed with a different model, and then some mitigation, looking at additives, extended storage, etc. And then we'll see if we've really made a difference in the end. What's our impact? Have we really helped the industry? So let's start out, first of all, with what's happening in China. Now, Pipestone, I think, has about 70,000 to 100,000 sows that they own and manage in China. We've got an active division led by Joseph Yaros and Gordon Spronk. So I went to those fellows and I asked them, hey, what's happening over there? Because in regards to North America, this is where we have to keep our eye on the ball. What's happening in China? And they said, well, here's a few things. First of all, the national herd really got hit by ASF in 2018 and follow-up years when the virus first got into the country. It went down from 50 million to 15 million, maybe, nobody really knows. Then they started to bring it back up again, but now that's slowed. There's been some issues that have slowed growth. And again, a lot of it's due to ASF. The reason why people want to be in business, in the pig business in China, is it's big bucks. You see the price of wean pigs, roughly $250 US with a $40 cost of production. Gilts, anything that will breed, whether it's got 3 legs or 4 legs or whatever, is going to $600, $700, $800 a gilt with US dollars, with maybe $100 cost of production. So it's very, very lucrative to be in the pig business. And that's why they don't really want to eliminate the virus. They don't want to come in and stamp out herds and drop production. So the important thing there is the wild virus is still circulating. And they've come up with some novel methods. It's kind of like a fancy test and removal called tooth extraction, where they'll go in, pull out an individual clinical animal, and an animal on either side in the breeding and gestation area, and try to eliminate virus that way. That's called tooth extraction, like you're pulling the bad tooth out of the mouth. That's used to work pretty good, but now with some of the issues that vaccines are providing, it's a little different story. Vaccines are now being used in China. They're modified live, they're gene deleted. There's several of them. They shed and they spread, and they're actually quite negative on sow performance. So they're not being used in sows, but if they get into the breeding herd, you can really reduce performance of your sows. They do cut mortality in finishing. So for example, if ASF kills 95% of your pigs, which it does, we've seen that, the vaccine might cut that down to 50%. So can you believe that? They'll take it from 95 down to 50%. So everybody wants to use a vaccine because at least you're not losing the whole population anymore, but still only 50% survival. That's a little different than what we're used to. The problem with these vaccines is they're very hard to detect, and they spread kind of under the covers, so to speak. They're hard to pick up. They really confound these elimination efforts on the tooth extraction side, because it's hard to pick up these vaccine viruses in farms. And we're talking feed today. The Chinese are very familiar with that topic. They consider it very significant. We found ASF DNA in the commercial feed system in China. They don't want to talk about it or publish it, but it's there. And what they've come up with is this plan. They call it cooking. So after pelleting, everything's pelleted in China. Instead of a short period of cooling the pellets, they'll actually keep heating them up for another 3 minutes at 85 degrees C. So they'll kind of try to cook the virus. So that's kind of what's happening with African swine fever in China. It's a real can of worms. So the reason why we're interested in what's happening in China is because the fact that, at least in the United States, we import grains from China, and we import soy, and we import corn, and we import a lot of things. And this is the reason why we get concerned, is this is how grains are dried in Asia. I call it the Trojan horse, because you can see that these grains could easily be contaminated with viruses, and those viruses could make their way into, be it North America, through this method. And the reason why that's also important is this table here. That's a compilation of work that I've done with South Dakota State and with Kansas State, with Megan Niederwerder, Eric Nelson, Diego Diel. This is looking at viral survival in feed ingredients, which are listed on the left. Ingredients that the US brings in from China, viruses on the top. Look at ASF as the second column over. And these are going through a simulation. It's going through a model, trying to mimic the movement of feed ingredients from Beijing to Des Moines, for example, or from Eastern Europe to Des Moines. And every time you see a red square there, that means the virus lived throughout the journey, the 37-day journey from Asia, the 30-day journey from Eastern Europe. And you look at ASF and you just read down the line there, a lot of red. It survived in our models in all the soy products. It survived in choline chloride. It survived in all the pet foods and the pork sausage casings. There's even in our complete feed positive control. And then the stock virus control, the very bottom line, that's actually the virus by itself in our model container in the absence of a feed matrix. And so this virus survives extremely well with or without the help of feed. And as you read across, you look at the various ingredients: soybean meal, conventional, which I'll call low fat, high protein; soybean meal organic, the opposite, high fat, low protein. Look at how well these viruses, not just ASF, but other viruses, Seneca, which is a surrogate for FMD, pseudorabies virus, PED, hog cholera, classical swine fever, and even PRRS, lives very well in soy-based products. And then in green, you see where the viruses didn't survive. So some viruses like influenza virus of swine, if you go all the way to the right side, IAVS, it didn't live in anything. So some viruses are very hardy and stable, and they live well in feed ingredients, others don't. Now, why is this important as far as imports to the US? These are data from work I did with Gil Patterson, looking at the country of origin, ASF positive countries that we are bringing feed ingredients that are soy-based into our country. And so you see Ukraine, Russia, and China, they're circled in 2018, 2019. Those were the top countries where we imported soy-based products into the United States. And so that's kind of scary because all of those countries are ASF positive. ASF, I just showed you, lives very well in soy. And so it kind of makes you, oh my gosh, this is a real risk. This is happening on a regular basis. And so that's just a quick review. So we know that certain viruses live very well in certain feed ingredients. Certain feed ingredients are very protective. And at least from the United States point of view, and I know you've studied this at a great deal in Canada, as we import products from high-risk countries, we have to do something about it. And we'll talk about that in a minute. Now, let's get into the new experiments. This is a new experiment that I just finished up. I call it evidence. Notice that word, we're going to use evidence in this case. A viral survival in feed and feed ingredients during a long-distance transport event across the continental United States. And you see the supporting cast of characters there that help me with this product. So this is a cool project because this is where we're going to take viruses and we're going to put them into a real-world transport event. And we're going to use 1-ton totes to inoculate with viruses: Seneca, PRRS, and PED. Of course, I can't work with ASF in these conditions, so we have to work with viruses that we can manage. So these studies will all involve PRRS, Seneca, which again is a great surrogate for FMD, and PED. So we're going to take 1-ton totes you see lined up there in our warehouse. We're going to spike him with a little ice cube. On the right hand, you see the 10 milliliter ice cube containing those 3 viruses, PRRS, PED, and Seneca, at 5 logs each. Okay. So what we're going to do is create a hotspot. So we're going to take one of the totes, could be soy, could be complete feed. We're going to hold it up above an empty tote. We're going to open the spout, as you see on the right side, and let the feed flow from the top to the bottom. Now, the reason why I wanted to do this was I wanted to kind of create a hotspot. With aflatoxin contamination of grain, aflatoxin is very focused as a hotspot. It's not widely dispersed throughout the tonnage. I wanted to do that. That's how I think this works with viruses, and I wanted to create the same thing. So you see the left picture, you can see the bottom tote is filling. So during the filling process, when the bottom tote was about half full, you see my arm there in the middle picture. I'm looking away, I'm throwing that ice cube, literally a 10 milliliter ice cube like you have in your drink, containing the viruses into the middle of the bag. And there on the right side, you can see what that looks like. I just stopped and took a photograph to show you the hot spot circled in red. Then we let the remainder of the upper tote fill. So I wanted to create this hot spot in a blinded fashion where I didn't know where it was. And I wanted to have it buried within the tote, so it'd be difficult to find. Now we're going to sample these totes using a grain probe, using a method that Cassie Jones has validated. I'll explain that in a minute. So there you see our probe, there you see our tote, you see in the middle there, the length of the probe in comparison to the tote. And then on the right, you see an interesting little, several little flags here. This is how we're going to sample the tote. We're going to take 10 grain probes. This is a standardized protocol by the feed control officials in America. We're going to take 10 samples from this one tote and make it into one composite sample. So everywhere you see a little red flag, we're going to take a sample of feed in our grain probe and pool it all together. Now, before we set this off and get this project rolling down the road, we're going to bulletproof it. We're going to do everything we can to protect the surrounding areas as we travel along the way with contaminated feed. So we're not going to have any other products on board. We're not going to make any stops en route other than for gas and a hotel. We have a designated driver just running this truck all by itself, nothing else on it. We put a GPS tracker on it. I ran this by our FDA Center for Vet Med and our USDA to let them know I was going to do this study before we started to get feedback if they were concerned. And then I sent a letter to all the state animal health officials in states where the truck would be staying overnight, explaining the study in advance. Again, seeking feedback, raising awareness, letting everybody know this is what we're going to do. And everybody was really positive about it. I was really excited. The FDA thought it was great. The USDA thought it was great. The state animal health officials were very, very positive. And all I needed to do really was just label the totes, and I'll show how we did that, and then barricade them within the trailer. So this is a letter you can see on the left. This is a letter that Shannon George from the FDA Center for Vet Med wrote to me, approving the product, giving me the specific language that you can see on the right that the totes needed to be labeled with. So this wasn't going to go into any animal production system at the end. It's just basically going to go into the research enterprise and not into the commercial system. But you can see the language I had to label those totes. And then the letter there, that was carried in the cab with the driver throughout the entire trip. So you can see my information there in case anything had gone wrong. And there we push the totes in. We're going to have 7 totes. We're going to have 2 of conventional soy, so high protein, low fat. We're going to have 2 of organic soy, so higher fat, lower protein. And we're going to have 2 complete feed totes. So 6 totes that will be inoculated with the ice cube. Then we're going to have 1 complete feed tote uninoculated as a control. That's the one you see right there in front. So these 7 totes are going to be put on this truck all by themselves, pushed to the front, and then barricaded with that door you see on the right to be sure that in case there's an accident, these totes aren't going to be rolling around, potentially spilling inside the truck. Just trying to be sure we do everything we can to protect the surrounding environment. Now, along the way, we're going to put some controls in place. All good studies need controls. So they're in a little basket you see with those orange caps. Those are 30-gram samples of soybean meal, conventional soybean meal, organic, and complete feed, either spiked with the viral mix or not. So negative controls, positive controls. And those orange caps, they're vented. And so the feed and the virus can feel the environment as the truck drives around. So we need those controls just in case all our totes come up negative. Did we actually have virus survival or not? On the right, you see a temperature and relative humidity gauge, and we're going to try to measure some of the environmental parameters in the tote itself, deep down inside the 1 ton tote. Okay, so here we are. We're at a commercial loading dock. We have a commercial vehicle there on the right, the big 18-wheeler's ready to roll. So again, trying to do this real world. Commercial truck, commercial feed quantities, a hotspot challenge model, and all sorts of controls in place. And we're going to send the truck on this route. We're going to start up in Minneapolis, Minnesota, there at letter A. We're going to drive down to Kansas City, letter B. We're going to head out west, go over to Denver, down to Albuquerque, New Mexico, Fort Worth, Texas, New Orleans, Jacksonville, up the eastern seaboard, and then into the Atlantic states, and then back to the Midwest by the Great Lakes. And the reason for doing this is twofold. This is a commercial route. So besides a commercial vehicle and a commercial feed quantity, I wanted a representative route of travel that a delivery would actually go on if this was a true delivery. This is an actual delivery. The second reason for this route that I liked it was I wanted to expose the viruses and the feeds to as many different climates in the continental United States as I could. And so here you can see we started cold, we went out west to dry, we went down to moist, the Gulf Coast and up the seaboard and such, really trying to expose these viruses to all different conditions. So, interesting thing about this route was it was about 10,800 kilometers. It took 23 days. We covered 29 states. And so we really tried to be diversified in this route. Let's see what we learned. Let's talk about some of the environmental readings. Let's look at some of the PCR data from the grain probes at day 0 and day 23 at the end of the journey. And then the main point is let's assess whether the virus has lived or not. Now, you remember, we've got 4 soy totes. You can't feed that soy to pigs, but you can evaluate it through bioassay. So we're going to take samples from these totes. We're going to inoculate them into pigs intramuscularly for PRRS, intranasal for Seneca, and orally for PED. Now, then the complete feed totes, we put them on purpose because we're going to feed those complete feed totes to pigs. So we're going to see whether natural feeding behavior can result in infection in parallel with the bioassay of the soy products. So we've got a couple different viability metrics going on side by side. Okay, so here are the temperature and percent relative humidity readings during the 23-day transport period. So the left-hand side, you see location of probe. We've got it in a SBMC, that's soybean meal conventional, in the tote, soybean meal organic right below it, in the tote. You can see that over the transport time, we collect a lot of data. These probes read about every 15 minutes. And then you can see the temperature, mean, max, and min, relative humidity, mean, max, and min. You can see what it was like inside those totes. And what's interesting to me is there was significant differences in temperature across the 2 totes, and as well as relative humidity across the 2 totes. And that was interesting to me with the relative humidity. The conventional soybean meal tote had a relative humidity reading on average of 66%. In contrast, the organic soybean meal tote had a much lower humidity on average. So there was some differences in these totes as far as the environmental parameters within the material. Now, when we sample with the grain probe, as you remember, we're going to pool the 10 samples into one. And that's what you see there up on the left-hand, upper left-hand corner is about a 1 kilogram composite is what came out of these probes. So all 10 together. And so the question was, how do you test that 1 kilogram? One way with, well, let's just take a little bit of it and see what we find. I didn't want to do that because what if we miss it? What if it's not distributed equally throughout the entire allotment? What if we wash it? So what if we fill it up with saline, as you see on the bottom there, on the bottom left, shake it real good, put it on a paint shaker, and let that run for like 2 minutes and really wash this entire amount to get exposure of the entire 1 kilogram. And what if we put that then into a centrifuge and we spin it down and we take the supernatant off those samples as our means to measure by PCR or by bioassay. So Aaron Singri and Eric Nelson from South Dakota State came up with this novel approach, and I think it really fit the bill. So what did we learn? So here are the PCR data from the totes. So on the left, you see the 7 totes that we went on the road. Soybean meal C-1, that's conventional tote 1, soybean meal conventional 2, soybean meal organic 1, soybean meal organic 2, complete feed 1, complete feed 2, and then our negative control. DPI is days post-inoculation, so we took a sample at the start of the trip, day 0, and then we took one at day 23, a few columns over, at the end of the trip. And what you see here, the viruses on the top, we found RNA fairly readily in the soy products. We could find evidence by PCR, again, RNA, of PED, Seneca, and PERS for the most part across most of the soy. Now you look at the soybean meal O-1, you skirt over to day 23, neg, we fanned on that one. We must have missed the spot. See all those negatives there. You can look down at the complete feed, it's not quite as successful. You see a lot of negatives on those complete feed totes, except for Seneca. Seneca seems to be living in everything, just like it always has. And so it's just kind of, we saw that before with our laboratory-based stuff. So and then there's our negative control at the bottom, all negative. So the good news was we could find the virus. Now here are the control tubes. These are those 30-gram samples in those little orange cap tubes. Look what a nice model this is. Day 23, we took those samples, we tested them. Look how repeatable. When you can test the entire 30 grams, look how repeatable this is. We can find all the viruses at pretty strong CTs, and then our negatives were negative. And so this gave me confidence that this is a wonderful model for looking at viral survival in feed at small scale. And that's why we're using large totes, but it helps back up the fact that, yeah, these viruses are present in these products. And it also gave me confidence that all the negative controls were negative. So meaning cross-contamination is not an issue for us now. Now, to the most important, viability. Okay, so there's our totes and our positive and negative controls on the left. DPI, again, is day 23. Viability assay, bioassay, we've got pigs in a pen. We're going to inject the preparation, as we showed you, after we wash and spin it down. Again, intranasal, intramuscular, and oral. And then you go down to the complete feed and you see, we're going to feed the complete feed to pigs. We can't do that with the soy because the pigs aren't going to eat that, but they will eat the complete feed. So that was a beauty of putting complete feed in this model, is we could test it by natural feeding behavior, which becomes more of a transmission study as well as a survival study. So the 3 viruses on top, and you kind of scroll down there, you see, yeah, we found live virus by bioassay. Several times we found live virus. Sometimes some samples were negative, like the PERS up in the upper right-hand corner, but sometimes the PERS was positive on bioassay, as was the PED and the Seneca. Notice that third line down, soybean meal O-1, that's the one that we whiffed on in the previous PCR slide. We just missed the spot. So all the bioassay samples were negative, which matched nicely with the PCR negatives. But look at how well the viruses survived in the treatments as well as in the controls. Now you go down to the complete feeding, about a second to the last line, we see we found evidence of viable PED and Seneca virus, but not PERS. I've got the PERS negative circled in green to highlight the fact that the PERS virus did not survive the 23-day journey in complete feed. It did in soy, as you see up above, but it did not in complete feed. And the bioassay, the positive control below that, matches that nicely. So that virus, as you know, is not as stable, it's not as hardy, it did not survive the journey. It did, though, in the soy, kind of showing you again the protective effects of soy. So this is like really important, because now we've shown for the first time evidence. That's why I use that term to start off the title, is I think this is more than data. This is more than just results and observations. This is a collective body of evidence that these viruses can survive in representative volumes of feed and feed ingredients under actual conditions of long-distance commercial transport across the continental US. We've never done this at this level before. It's always been in the lab. Now we've taken it out of the lab into the real world, really pushed the envelope, and clearly shown that these viruses can survive under these conditions. So with that in mind, it raises the question, well, what are we going to do about that? That's a major concern. And you all know that from Canada. That's why you've got your national program in place. But I wanted to then evaluate feed additives as a means to reduce the risk of viral survival in contaminated feed. Now, I did this project at our BSL-2 facility in Pipestone. You see, it was about a 1.5 year long study. Our BSL-2 is nice because it's 6 rooms. Each room has its own bin. There's separate air spaces, so we can have treatments and controls side by side. We can have treated feed in one bin and non-treated feed in another bin. We can take different products and mix them up in individual bins and feed them to pigs and see what happens. Do the additives that we put in the treated feed have an effect on killing the virus, or do the virus just continue to survive? And remember, we'll always have a control group, which is a room of non-mitigated feed. So 96 pigs in a room, designated feed, treatment, and control. Here are the companies and the products that we tested. We put out a call for participants for this study. Bring your product, bring a set amount of funds, and let's let it roll. And so you see the companies there on the left who all participated, the various products that were tested under this in this experiment. Several different chemistries is what I really liked about this. We had acids, we had formaldehyde, we had medium chain, we had long and short chain fatty acid, we had blends, we had essential oils, we had probiotic fibers. We had a lot of different chemistry, which I was excited about. 2 products to note. The Alltech Guardian there with the red asterisk, I developed that product. I have a conflict of interest. So just for transparency, I'm disclosing that. Provimi Vigilex, this is a product that they wanted to test it, then they pulled out. I'm not sure why, but some producer colleagues of ours wanted it tested because they were using it. And so the producers paid for the Vigilex fees. Everybody else brought their own money, but the producers stood up and said, well, let's see how it works. So that was pretty nice. Now the challenge, again, we're going to use our 3 viruses, PERS, Seneca, and PD. My idea was, let's make a block of ice and let's put equal amounts of each virus in. You know, we talked about ice cubes a study ago. Now we're actually going to talk about a block of ice that weighs 454 grams. It's made up of all 3 of those viruses, again at 5 logs TCID50, frozen at -80 in Eric Nelson's lab. And then basically we're going to drop those blocks into the bins of treated or non-treated feed at day 0 and day 6 of the project period. And then Mother Nature takes over and the blocks melt or they chip up by the auger and the feed moves into the room, the pigs eat it. So here's a picture of the block sitting in the middle of the feed after it's been dropped. And now in the barn, in each room, we're going to measure several different ways. We're going to take Swiffer samples of the feeders, as you can see in the upper right, to see whether the virus got into the feeder. Did we actually find evidence of the virus in the feed at the feeder level? Remember, we threw it in the bin. Did it make it in the room? Oral fluids we collected to see whether the pigs contacted the virus by eating. Then we looked at clinical scores. Did the pigs get sick? Post-mortem, we collected, of each 96 pig room, we collected 30 pigs at the end of the study and looked at tonsil samples to see whether they were infected with Seneca, looked at serum to see whether we could document PERS infection, and then looked at rectal swabs to see if we could find PD infection. And then we measured performance, and that's what I'm going to highlight for you in the next slide, but that's what really counts is, do the pigs do better when they're on mitigated diets versus non-mitigated diets? And I'm just going to show you one experiment. We ran 5 different experiments, but just to give you a feeling for what we saw, each experiment, you see we have in this case, there were 5 products. You see the inclusion rates in parentheses, and then you see the positive control. So again, these are the 6 rooms of the building. Each room has its own feed bin with its own product, except for the control. And you just hop over to the middle column, look at average daily gain. You can see that 4 of the 5 mitigants seem to have a positive effect on gain. The 5th mitigant, the Vigilex product, it was no better than the control. And then you see the mortality. very different on the first 4 products as compared to the Vigilex and the positive control. In combination across all 5 experiments, this is a nice meta-analysis our biostatistician Roy Edler did, just looking at if we take the average daily gain data across each experiment, treatment versus control, does it benefit to have a mitigant in place? You go over to the right side of the slide, you see that forest plot, you see the 0 bar, you see those black symbols to the right of the 0 bar. That's indicating there is a beneficial effect of using a mitigant on average daily gain. If the black symbols are on the other side, it would say, no, the controls, there's no benefit at all. They're no different than the controls. So overall, you can see across all 5 of the experiments, yeah, mitigation was a good thing to do. So as we concluded that study, we saw improved health and performance in the face of infection on mitigated diets. 14 of the 15 products we tested provided significantly improved performance as compared to the control. The one did not, I just showed you. So what was really cool was you had pigs with evidence of the virus in their bodies, either by blood or tonsil or rectal swab, yet they were growing like weeds. They were really, really growing. They were healthy. They did not die. They handled the challenge very well. And then I like this about at the end of it all because we had diverse chemistries, a lot of different products, a lot of different options. That's good for producers and for millers. So there's not one size never fits all in everything. So I was happy about that. Last experiment is a holding time experiment. And I think this is a new one. I'm not sure if you've seen this one before. This is really looking at a single protocol of extended storage as a way to reduce viral risk. So we're going to kind of use some of the same methods I've talked about. So briefly, we'll just say, hey, we're going to have 1-ton totes. They're going to get inoculated with our ice cube per Seneca PD. They're going to sit in a warehouse at 20 degrees C for a 35-day period of time. We're also going to have a control group down below, you see, of totes inoculated with the ice cube sitting in a tractor shed on a farm in Minnesota. So not a climate controlled environment as compared to the holding time environment above. So what did we learn? We measured temperature in the totes. I'm sorry, this is the environment. The internal environment, you see, it was roughly 20 degrees, 19 to 21 degrees, very stable. The external totes, not the case. We had much colder temperatures. This was done in January in Minnesota. So you can see there was quite a difference in the environments between the stored bags inside and the stored bags outside. Here's the PCR data we sampled with the grain probe, same approach as last time. So you can see we had 2 totes of soybean meal conventional, we had 2 totes of organic, we had 2 totes of lysine in this case as well. So 6 total totes, today's day 0 versus day 35, which was our storage period. And the question was, could we find the RNA? You can see in this case, sometimes we missed it. There was lots of negative samples here. The grain probe sampling is a hit and miss deal. Remember, we don't know where the cube is. It's buried somewhere in the middle of this 1-ton tote. We're basically just following that little pattern of taking 10 samples and hoping that we find it. But in the 30-35, we got a little bit more, maybe a little more luck involved. We found evidence of the RNA. Those were the internal, here's the external storage totes. Lots of this one, we hit the jackpot. We found a lot of positive samples in these totes that were held in the farm shed in Minnesota in December or January. Sorry. Here's our controls. We had some of those as well, those little tubes with the orange caps. Again, a very nice model showing that if you can measure or evaluate the entire 30 grams, you'll probably find the virus pretty nicely. And it also showed again that our negatives were negative, again, showing that we don't have cross-contamination going on. Now for viability, we basically ran bioassays. And so we took the internal totes and we processed the samples like we talked about on the paint shaker. We inoculated them to pigs in our BSL-2. You can see no virus survived. All viruses for the 35-day period at 20 degrees C, no viruses survived that protocol. In contrast, the outside totes, all the viruses survived. So it was kind of a black and white outcome as expected. Viruses always like cold temperatures. They don't like warm temperatures. And so this made a lot of sense. So at least for this one protocol, This is only one protocol. Again, 20 degrees C for 35 days, effectively inactivated important viruses of swine. And then based on these results, remember the external and the internal samples were both held at 35 days, yet we found viable virus in the external sample. So basically it's saying time is not the major factor. Temperature is more critical than time. So let's close up and then we'll see if they have any questions when it's available for question period. But I think there's a few points here we have to really make sure everyone understands. Is there's a lot of information now. There's a growing body of scientific evidence that certain feed ingredients can support the transport and transmission of multiple viral pathogens. This has been done ad nauseum. It's been done across multiple universities, multiple laboratories. You saw the large real world study we did. I don't think we need to do any more of these. I think there's enough information that says feed is a risk. We now got, I think, some options to validate that have been validated so we can mitigate the risk, holding time, additives. And overall, I think this has really changed human behavior at the level of the vet profession and the swine industry. You know, it's kind of like having health papers for feed now. As feed enters a country, say in Canada, it has to have certification. Certain information has to accompany it, just like a health paper for animals. We never used to do that before. Then we put the feed into a holding where we treat it, you know, almost like a quarantine. That never used to happen before. This has changed human behavior. And I commend once again your country and Australia for taking the lead and putting national programs in place to manage this risk. So thank you and congratulations. And my last slide is to bring back some people who left us too early. There's a great Canadian there. There's Bob Morrison. And there's a great American, that's Al Lehman. And you see a great Mexican, Carlos Pijuan. Unfortunately, all three have passed. However, if they were here today, I guarantee they would be pounding the podium saying it's time for a North American program. Canada can't do this by themselves. If the US and Mexico aren't following the rules too, it's a wash. It's not a good thing. And so we have to think about this. And that's my message I want to leave you with, is we have to work together to get this done at the continental level. And we are having conversations with Canada, with Mexico, and the United States to try to see what we can do about that. So with that, Dr. O'Sullivan, that's my presentation. I do appreciate the invitation to the London Swine Conference. I hope you found it interesting with some new information, and thank you very much.
Thank you, Dr. D. Really interesting work and very impressive work. And I really enjoyed sincerely listening to that. And I feel quite lucky to have had my own personal private viewings. So thank you. And I look forward to hearing more about your future research results.
Thank you very much.
Thanks. And so that brings this presentation to a close, everyone. And once again, on behalf of the London Swine Conference, I would like to thank you for visiting this presentation and also thank our sponsors that have helped make the London Swine Conference possible this year. Thank you, everyone.